So, greetings, ladies and gentlemen. What we're going to talk about today is um, the mechanisms of and effects of mutations as well as gene transfer. And one of the end results of all of this is genetic variation, which is important because otherwise we'd all look, act, and react exactly the same way, which would be bad if any changes to our environment were to occur. So that's why you are able to look differently from anyone else because long ago there was some mutation in a gene. Okay. So, what is a mutation? A mutation is any change in a DNA sequence, and this can happen at multiple levels. It can happen at the level of a nucleotide, so you have one nucleotide changed in a gene, or it can involve entire segments of chromosomes. So we'll talk about both these kind of small level mutations as well as the larger level chromosomal mutations. What are the results? Well, you could have something that does not change any sort of physical characteristic. You could have a change in the type of protein that's produced, so you could have a change in the structure. You could have a change in the amount of protein that's produced, and that will change the trait. So you have to have a change in one of those. You have to have a change in the amino acid sequence or the amount of protein expressed, or even where the protein is expressed to affect the phenotype of an organism. And all the different phenotypes that we see ultimately arose from some sort of mutation. So let's look at a very dramatic example of a mutation. So what you're looking at is the primary structure right here of hemoglobin. Hemoglobin is the molecule that completely fills up your red blood cells and also is what carries oxygen around inside your body. And so what happens in some parts of the population is something happens to this guy right here, the sixth amino acid in the primary sequence, glutamate. That glutamate in some individuals was accidentally changed to a valine. Basically, the DNA changed. With the DNA changing, the amino acid it coded for changed, and with the amino acid changing, we had a kind of large scale change in the structure of this protein. So it happened to be in such a place and it happened to be such a change that basically it changed the whole shape of this subunit of hemoglobin. So hemoglobin has four different units, two alphas, two betas. Well, it changes the shape of the beta subunit. Basically, it's got this hydrophobic region then exposed to the surrounding fluid. Remember, hydrophobic areas on any molecules are going to be rejected and kind of clump up, which ends up causing these beta subunits to kind of join their hydrophobic region so that way they're not exposed to water. Water excludes them. And that causes these hemoglobin subunits to kind of form this large crystal or a fiber. That not only reduces its ability to carry oxygen, it also results in the property for which the disease is named, which is the particular shape of the red blood cells. They end up getting changed into this sickle shape. That's like a sickle. It was an old school tool used to kind of reap wheat and everything. And these sickle shaped cells have more trouble fitting through capillaries because capillaries are so small, red blood cells have to go through them single file. Well, you could get some of those sickle cells basically stuck trying to get into a capillary, and then you would block the capillary. And that's responsible for a lot of the organism level symptoms that you see of sickle cell anemia. So, this one change in this one amino acid leads to changes all the way throughout the body that end up causing a disease that without modern medicine would end up being fatal. People with sickle cell anemia need frequent blood transfusions because they're not producing red blood cells that are round. So then we can replace their red blood cells that are sickle shaped with round red blood cells. But without that, throughout their lives, they wouldn't survive. So this is just showing you how a tiny change can end up making a big difference in an organism. So where do mutations ultimately come from? There are three major sources, one of which is, uh, well, there are actually more than three major sources. So inside your body, there's one major source. You make an error when you're copying your DNA, or you make a mistake when you're repairing DNA. And this just happens naturally. It's not super rare. Luckily, you do have plenty of enzymes whose job it is to go through your DNA and try to fix mistakes. But like anything in the universe, mistakes happen. So that can be the source of a mutation. You can also have three major external factors. Radiation. So anything that gives you UV radiation can give you a mutation, a change in your DNA. X-rays. This is why, for example, like if you've had there's a lifetime level of radiation dosage you need to stay below in order to, ward off risks of certain cancers. The higher the radiation doses you get, the more likely you are to get cancers. Because of this, if you're like an X-ray tech, you have to make sure that you are protected from any of the X-rays that you are shining onto your patient. Otherwise, it increases your risk of mutations, which unfortunately most of the time increases your risk of cancer. There are also chemicals, which you can call either mutagens or, in a lot of cases, carcinogens, that can cause mutations. Carcinogens are especially known to cause cancer. That's what they're named for. Mutagens are just anything that can cause changes in DNA. And then you can also have infectious agents. Most commonly, viruses are especially bad at causing mutations because they insert their genetic material into the host genetic material, which, if they're infecting you, could cause a mutation in your genes. Now, most of these mutations, unless it's like an error during DNA replication in cells that are going to make gametes, so sperm or eggs, most of these would just affect the individual who has that mutation happening. If you want to have a mutation that's passed on to the next generation, it needs to occur in the sperm or in the eggs, which is possible. Um, so let's look at what happens with mutations. Mutations are not good or bad. They're just changes in DNA. However, they can have positive effects. They can make a protein work more efficiently. They can make a process more efficient. They can make some sort of structural change that would benefit the organism. They could be negative. They could completely destroy the function of a protein or make it less efficient or cause less of a necessary protein to be produced, or they could be neutral. Neutral is usually either something called a silent mutation, where a nucleotide sequence is changed, but it doesn't end up changing the amino acid sequence. And if you're wondering how that's possible, we're going to take a more detailed look at that in a moment. Or a neutral mutation could just change something in a part of the protein that doesn't dramatically affect its structure, doesn't dramatically affect maybe the active site if it's an enzyme, or it could be that you changed one amino acid for another one with a similar property, like you exchanged a hydrophobic amino acid for another hydrophobic amino acid. Ultimately, the mutations that survive in a population can ultimately become new alleles that can be inherited by individuals, and this ultimately increases genetic variation, which is going to tie in very closely to when we talk about evolution, because evolution is only possible because of genetic variation. If you don't have variation in the population, then you're not going to react differently to changes in the environment, and you're not going to have differential reproductive success, which is the hallmark of evolution. In general, it's better when you have high genetic variation. Now, it's not necessarily better, like, you shouldn't go out and irradiate a bunch of animals trying to give them lots of genetic variation. Um, Generally, populations with higher genetic variation are healthier and more likely to survive any changes in their environment just because they've got a large selection of traits that could help them out. Please don't go irradiate random populations of animals. Most of what you're likely to do is give them cancer. Not a good thing. So let's now talk about these actual types of mutations. So point mutations are usually single nucleotide mutations. We're going to focus on substitutions right now. So I said we were going to talk about silent mutations, and these don't affect either the phenotype or the final 3D structure of the protein. They don't even affect the amino acid sequence of it. So if we go back to our nice handy codon chart that tells us what amino acid is coded for by what codon in mRNA, we'll be able to see why this is the case. So we have DNA sequences. The first codon right here is TAT. If we were looking at what RNA would be produced from that, it should be AUA. If we look at AUA, maybe, oh, never mind. Sorry. I thought we were looking at the, I think we were looking at this is the coding strand, not the non-coding strand. Never mind. So TAT would actually be UAU if you were looking at the mRNA version. If you look at UAU, that codes for tyrosine, which is what we get. If that last nucleotide in the codon is replaced with a C, you would get UAC. Well, UAC also codes for tyrosine. This is due to the fact that you only have about 20 amino acids that you're coding for with this, but you have 64 different codons. So there is what we call redundancy in the genetic code. More than one codon can code for the same exact amino acids. So for example, you can have 4 different codons code for serine. Notice these guys all code for serine. If you change the last base in the codon, doesn't matter. You're still going to get the same amino acid. That's a good thing because that means that, these inevitable mutations that occur don't necessarily have to change the sequence of the protein. This is a silent mutation. On the other hand, you can have other types of mutations called nonsense mutations. They are going to change a codon so that instead of coding for an amino acid, it's going to transform into one of the 3 stop codons. And if you remember from the translation video, stop codon is basically going to direct a ribosome to dissociate, separate from the mRNA that's made, and you're going to stop making protein. So that's like your stop sign. Okay, you're done. It's like the period at the end of a sentence. You're done, move on to the next sentence. You're done, move on to the next protein. So here we have a point mutation where instead of UAC, it changed to UAA. Well, UAA, which is over here, is a stop codon. So instead of continuing to make more of the protein, the ribosome just stops. It's just following instructions. Remember, these are all molecules. They do what their function dictates, but they don't have brains to think, wait, this shouldn't be the end of the protein. So instead of making a protein with, say, 300 amino acids in it, depending on where this mutation happens, you could have 10 amino acids, 50 amino acids. If this happens more towards the end of a protein, let's say that this is your protein right here. If your mutation happens over here, and that's a premature stop codon put in there, probably not going to affect the function of your protein as much. On the other hand, if your premature stop codon is somewhere near the middle or in the end, you can just basically make a non-functional protein. So that's what you see right here. You see this yellow protein is the full-size normal protein when it's all folded up. The produced protein with the nonsense mutation, though, is shorter. A missense mutation is a little more complicated. So a missense mutation is going to basically change, most of the time, it's going to change, from one amino acid to another. So it alters a codon so that instead of, say, here, where this codon is coding for glycine, instead, you're going to change it so it codes for alanine instead. Now, if the amino acid that's substituted is very similar in its chemical properties, then it may cause very little change to the structure of the protein, and then very little change to the function of the protein. And, these may be like loss of function, where you have less protein produced, or your protein is less effective, or they could be gain of function mutations, where the resulting protein works a little better. On the other hand, what's more likely is that you're going to have an amino acid substituted that has different chemical properties. I don't remember off the top of my head the properties of glycine and alanine, but let's say one of these was polar, one of these was nonpolar. I believe glycine would be nonpolar. We'll pretend like alanine is polar. Well, if you substitute a polar for a nonpolar amino acid, that's going to affect how this protein folds in its secondary, well, actually, its tertiary structure, because, the hydrophobic parts of a protein tend to be towards the center of the protein. So notice we have this kind of green dot right here representing the mutation. And notice that our normal protein, it's the same length, but it looks different in its 3D structure. If the 3D structure changes enough, then the protein will not be functional. So, and it kind of depends on where in the protein the mutation is located as well. So for this, I can't make generalizations. It depends on the mutation. It depends on the final protein. And it depends on the function of the original protein, what happens. So this is just kind of showing you what's going on. So this is our original DNA sequence. Here's the mRNA that is formed from it. If you change, for example, this one letter, that is going to cause a silent mutation because both AAG and AAA code for lysine. On the other hand, if you changed the first letter of that codon, you could create a premature stop codon, which means it's a nonsense mutation. And then you can have missense mutations, where you change a different base in the codon. And for example, here, you don't need to know the difference between conservative and non-conservative. We're not going to go over that. But just know that this conservative is showing arginine has a similar structure. So it's showing you the structure of arginine versus lysine. Arginine has similar properties. It is polar, NH2. Lysine has an NH3+. They both have a positive charge. And so that will likely have some similarities in its chemical properties, may not change the shape of the protein. On the other hand, if you changed it, for example, to threonine, which looks like it's mostly nonpolar and it's definitely uncharged, then you're going to have different, oh, wait, no, this is basic. Sorry. Basic, 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 and polar. So they're going to have different chemical properties. They're going to interact differently with other side chains of the different amino acids, and they could cause the protein to fold differently, which means it may not work as well. There's something else called a frameshift mutation. It's not so much a point mutation because what you end up doing is you end up taking out or inserting extra letters. So let's look at, and this is due to the fact that if you look at this piece of DNA, there is no physical separation between codons on there. It's not like in sentences where you have a space to separate words. Instead, what ends up happening, let's say you have an RNA polymerase. Let's say this is my RNA polymerase. It's going to basically read, it's going to have 3 bases sticking out. This is actually more a ribosome, but we're gonna go with it. Let's say that's AAA, and those 3 bases are just gonna kind of read DNA 3 bases at a time. However, if I moved things over, let's say this is UUU. And then before that was a C, and after that was a G. Well, if I moved that along, whatever 3 that's being read here, and I had them read instead of AUG, there was CUUUG. Now, instead of reading a UUU, they're going to read a UUG. Everything got shifted over by 1. So let's look at that happen up here. Here we have a wild type gene. This is the mRNA that's made from it. It starts out with AUG, which happens to be what all proteins start out with, which is going to give you the sequence methionine, lysine, phenylalanine, glycine. Okay. Then you look at this frameshift mutation. Instead of this DNA sequence being TAC, TTC, an extra A got stuck in there. That extra A changes the sequence of the mRNA. So now you still have methionine before that, but after that change, downstream, everything else is different. So, for example, you start out with methionine, but then you end up getting a nonsense mutation because the next codon ends up becoming a stop codon. Then you could also have a frame shift that results in a missense mutation. That's what we see down here at the bottom, where we had a deletion instead. So this deletion from right here ended up taking out a single nitrogen base. And then when this was divided up into codons by the tRNAs in the ribosome, instead of reading a UUU here, they read a UUG, which changed it from a phenylalanine, which is what this UUU was, to a leucine. But then everything after that also got shifted over. So next, it's going to read GCU instead of GGC, which is an alanine. And then every single amino acid after that is going to change. So a frameshift mutation doesn't cause a change in a single amino acid. It can cause a change in most of the amino acids. So that's something to keep in mind. Frameshift mutations can be much more dramatic of a mutation because they can affect most of the length of a protein. So here we see some different mutations that lead to kind of similar diseases. So these are various mutations that cause a disease called cystic fibrosis. Cystic fibrosis ends up with the disease at the person level being they make excess mucus. It is fatal usually without medical treatment. Luckily, medical treatment has improved. And so one area of research has been, what is causing this? What mutations? Well, this is kind of the normal pathway of endoplasmic reticulum to Golgi to the actual um, ion channel that's involved in this. It's a misfunction in a calcium ion channel being inserted into the membrane of cells in your respiratory system. Here, you've got a certain type of mutation that makes you not produce this channel at all. So nothing's being produced. Here, you have 3 different types of mutations that cause that. So you can have these 3 different mutations have the same effect because all of them result in not having that protein produced. Next, there's another place where instead of sending, so some of it got sent to this, looked like a proteasome. Sorry, the quality is bad. It was the best image I could find. So instead of some getting sent off here, all of it's getting sent over here to the Golgi, and it's just not targeted to the right place. That can happen through a single amino acid deletion or all of these different 4 missense mutations. Then you could also have a channel that's produced but is not able to function properly, so it ends up in the right place. It just doesn't work as well. And there are 1, 2, 3, 4, 5, 6 different missense mutations that cause that. So that's not like the entire protein is deleted. There's a mistake, there's a change somewhere where it is important to the structure of that protein. Then you have other missense mutations that cause the protein to not work as well. Others that will reduce the likelihood that it will be transcribed and synthesized. But all of these end up resulting in the same disease because they all result in the same effect, meaning that you have a misfunctioning calcium chloride channel, not chloride, calcium chloride, chloride channel, and then that causes the disease cystic fibrosis. This is why it can be really difficult to figure out, okay, you have, let's say, type 1 diabetes. What's the genetic problem that gives you this? Well, there's multiple ways that you could have that. So it's not as easy to pinpoint the cause of a disease or even how to treat a disease. Here, this is just kind of a word examples showing you how these different mutations can affect it and change the meaning. So, for example, you start out with beast. If you changed and substituted one letter, the first letter from a B to an F, that would give you feast, which is a totally different meaning. If you added an extra letter after the first one, that would give you breast, which is a different meaning from beast. If you deleted the A from the middle, then that would give you best instead of beast, which means something totally different. And an inversion, when you invert something, you basically flip it around. So here we have an inversion where our T and our S have changed places, and instead of beast, we get beats. So that's just showing you how you can change the order of things, or mess with the letters in a word, and it changes the meaning. Same thing happens in a person's genome. If you change or upset the order of the nucleic of the nitrogen bases in a gene, you can completely change the meaning of or function of the protein that's made. So now let's take a look at some chromosomal mutations. All right, so these end up happening when chromosomes break, and then instead of rejoining in the proper place, they rejoin to the wrong chromosome or wrong part of the chromosome. This is going to commonly happen during crossing over because that's a period of time when you're going to have a lot of breaks happening in chromosomes. Usually, what's supposed to happen is a chunk of chromosome will attach to its homologous pair. Sometimes things go wrong, though. And you have 4 different types of chromosomal mutations, which we'll look at each in their own detail. A deletion is where a chunk of a chromosome gets just completely removed. If this happens to a large enough part of a chromosome, this could be fatal. If this happened in a gamete, then this could produce an offspring or a zygote that's not able to develop till term, which would result in a miscarriage. However, if it's a small enough section of DNA, you might be able to get by without it. It doesn't have very many genes in it. Here we have a duplication, and that's where instead of having, basically you get part of it's like somebody did a copy and paste on part of the chromosome and added an extra duplicate of some other part of the chromosome. Once again, depending on how much of the chromosome is duplicated, if there's not very much duplicated, it could be harmless. However, a lot of genetics is about balance. So if you have more than 2 copies of a gene, let's say you have 3 copies of like 5 genes, that can cause some serious problems. Everything works best when you're producing things so that you have 2 copies of every gene. And we're going to look more in detail in that in a moment. Inversion is when a piece of chromosome breaks off and it reattaches to the right chromosome, but it does it upside down or backwards, so it's inverted. And if the genes can't be read properly in this, or if a break happens in an important gene, that can be very harmful or fatal. An insertion is when you add a gene from somewhere else, from some totally different chromosome. So that's what we see here happening. A gene from a totally different chromosome, gene 9, has suddenly been inserted. If it comes into the middle of a gene, it could make it non-functional. And a translocation is basically where you have 2 chromosomes swap parts. So here we see our purple chromosome and our green chromosome have switched some parts. They are not homologous though, so they're not like exchanging the exact same regions, which is why depending on where this translocation happens, it could, if it interrupts a crucial gene, be lethal or extremely harmful. There are also some mutations caused by errors in mitosis and meiosis. The most common one is nondisjunction, and that is when homologous chromosomes don't separate during meiosis 1, or you can also have it happen when sister chromatids don't separate during meiosis 2. The end result is you have 2 copies of a particular chromosome in one gamete, or you could have 0 copies of a particular chromosome in another gamete. And depending on what gamete is fertilized or fertilizes another gamete, this could result in something called trisomy. Trisomy literally means 3 bodies. And this is when you have 3 copies of a chromosome instead of 2. There's one really well-known trisomy of chromosome 21, which is Down syndrome. One is only you only have people able to survive, and they survive into adulthood with Down syndrome because it's a very small chromosome. There's a couple of other trisomies, but usually the infants who are born with them don't survive at past childhood, just because the kind of physical effects are so dramatic that it's not possible for the person to survive. So that's why Down syndrome is the most well known. So here we see what's called the karyotype of an individual with Down syndrome. We're looking at all of their chromosomes mapped out. And if you're looking for an abnormality, that's the abnormality there. So there are 3 copies of chromosome 21. Notice how tiny chromosome 21 is compared to all the others. That's the only reason why a person with trisomy 21 can actually survive, because it's a tiny amount of DNA that's been duplicated. People with Down syndrome, they're going to be born in approximately every 800 to 1,000 births, so it's fairly common in the population. The chance of having a child with Down syndrome increases with the age of the mother. So closer to 50 you get, having a child, if you're a woman, the more likely that the child will have Down syndrome. There are certain physical characteristics that are common to people with Down syndrome, and there are also some learning disabilities as well. It depends on the severity of the Down syndrome. Some people with Down syndrome are able to live independently. Others may need to have a caretaker or have uh, like uh, live-in assistance. Another thing that can happen is if you have some sort of event happen where you don't have any of your homologous chromosomes separated at all. Let's say if you had a diploid gamete fertilizing a haploid gamete, that would actually create something called a triploid organism. Triploid organisms have 3 sets of chromosomes instead of 1 or 2, and they are sterile. They're not capable of reproducing. You could have other circumstances where a diploid gamete fertilizes another diploid gamete. That would produce a tetraploid organism with 4 sets of chromosomes. These organisms are capable of reproducing, but only with other tetraploid organisms of the same species. They can't reproduce with a diploid organism because if they did, the tetraploids would make gametes with 2 sets of chromosomes. The diploids would make gametes with 1 set of chromosomes, and they would make triploid offspring. So they can't, nothing can happen there. There are very few tetraploid animals. There's like 1 species of frog that I found out. And it lives near and is very similar to the species it originated from, but they can't interbreed because they have different numbers of chromosomes. You can have polyploidy, which is more than 2 sets of chromosomes. You can have triploid, tetraploid. My favorite one to say for some reason is octoploid, which is 8 sets of chromosomes. And while that is not gonna, I've never heard of anything above a tetraploid animal. Um, Polyploid plants can be very common. And they are capable of reproducing. So, one of the effects of having polyploid plants is they tend to produce larger structures, larger flowers, and what we find relevant, larger fruits and vegetables. So, what you're seeing is a diploid strawberry on the top, notice the size, and a much larger octoploid strawberry on the bottom. So, most of the plants that we tend to eat nowadays are polyploid. So let's talk about how genetic information gets transferred. There are 2 major mechanisms. Vertical transfer is the type that we've been talking about, and that is when parents pass their genes down to their offspring. That's going to produce completely new organisms, and it's going to create genetic variation if it's sexual reproduction. Horizontal gene transfer, though, happens when 2 individuals can exchange segments of DNA or donate segments of DNA, but they do not make brand new organisms. This is not something that's very common in eukaryotes. It is most commonly found in bacteria and prokaryotes. So, this is a way that you can increase genetic variation and transfer helpful traits without sexual reproduction. It's a common way that bacteria and other prokaryotes will increase their genetic variation because they don't perform sexual reproduction. All of their reproduction is asexual, so this is one way for them to get some genetic variability. There are four major mechanisms by which this happens. We're going to focus most on one of them, but basically, transformation is when these prokaryotes take up just naked DNA from their surroundings. Doesn't happen under all circumstances. And then transduction is when a virus transmits genetic information because viral DNA gets inserted into the host DNA. Conjugation is transfer of small packets of DNA from cell to cell. And transposition is, you can think of it like a jumping bean sort of gene or section of DNA. It moves and kind of jump around between either different segments of a chromosome or between different chromosomes even. So let's take a look at each of these. So transformation, we do this a lot in a lab. You can do what you call making bacterial cells competent, capable of taking up DNA. And if you have DNA around them, either fragments like what you see in part A of this diagram, or a plasmid, which we'll talk about in a moment, like you see in part B of this diagram, you can cause that bacterium to take it up. If you have that genetic material integrate into the host cell, then it could cause a transformed bacterium that you're able to then use for experimentation. It's a little less likely though, because if it doesn't get incorporated, since it's a little linear stretch of DNA, bacteria have defenses against that because usually it's a sign of viral infection and they'll destroy it. However, if you insert genes in a plasmid, plasmids are small circular bits of DNA, and they tend to stay pretty stable. Um, Transduction is a virus, so you can do this in a lab. Put the gene of your choice into a virus, have that virus infect a cell, and then that virus will inject its DNA into the host cell where it'll be incorporated. This is a good way to try to um, introduce genes into eukaryotes. The only thing is it's kind of hit or miss. You can't specify where the virus inserts its genetic information. So it could end up causing more problems because it could interrupt an important gene. Bacterial conjugation is also sometimes thought of as their major form of sexual reproduction because it's the most common way that they exchange bits of genetic information. So what they do is one bacterium will extend a pilus to another. It forms a little tube, which is basically a hollow tube with some cytoplasm in it between two bacteria, and they will make a copy of a plasmid that's found inside of them and send that copy into the recipient bacterium. That plasmid has any number of genes on it, and that those genes can then benefit that bacterium. Then once a bacterium has a plasmid, it can transmit it to any other bacterium that it is compatible with. So what are plasmids? These are small circular bits of DNA that are not part of the main bacterial chromosome. They can carry, 50, 100 genes, and they're usually not going to contain the genes that are essential for survival. They're kind of like extras or upgrades. So one very common one that we tend to be focused on is there are some genes that that give bacteria antibiotic resistance, and they can actually transmit this between them. So you could have some bacteria that aren't descended from, a common ancestor, or common, they're not like exact clones of each other, but they're the same species. And you can have one of them have a plasmid that has a gene for antibiotic resistance, and it can spread that throughout the population even without having to take the time to reproduce, which is why antibiotic resistance is such an issue because it can spread so quickly. These plasmids replicate on their own, so they're not dependent on the bacterium reproducing to make more copies. And then just to compare, a bacterial chromosome is much bigger, has around 4 million base pairs, anywhere from 3,000 on up genes. We're just doing like an average. A plasmid is much smaller, anywhere from 2,000 to 200,000 base pairs, and it's maybe 10 to 15 10 to 50 genes. And plasmids are really important in the lab because you can use them to add genes to bacteria so that way you can get them to produce proteins and do genetic research. And they can really rapidly transfer traits around within a bacterial population. Transposition is usually happening in eukaryotes. A transposon is called a jumping gene, and it basically is a segment of DNA that can very easily jump from one area to another. And you don't need to know anything about how that happens or anything like that. However, these transposable elements can move around in the genome. They can, cause mutations if they land in the middle of a gene, and you can also use them in the lab to try and artificially add some genes. All right, so that is the end of this lesson, and the next lesson will be biotechnology, our last one for this unit.